And then we can go now to tSNE. And tSNE UMAP are very similar. So most of the things that I will talk about tSNE will also be valid for UMAP with just a small differences. So tSNE is a graph-based and nonlinear dimensionality reduction technique. But imagine that you have, for example, the samples that are distributed in this manifold. So this is not a linear problem. So imagine that you have these dot here, the white one and this other one. So the distance between those two dots, if you consider, for example, a PCA, would be just the Euclidean distance between them. But if you consider the tSNE, we'll find the closest neighbors to this cell first, and then the closest neighbors to that cell, the closest neighbors to that cell, and so on and so forth, until you get to this cell here. So this will be the distance in the tSNE space. And that will differ a lot how you see the resolution of your data, how you can visualize that. And that's what we mean when we say that this is a non-linear dimensionality reduction technique. So in other words, tSNE calculates the distance based on the distances of the neighbor cells. But how does tSNE finds what is the best embedding? So imagine that you have your data on a higher dimension. This representing all the dimensions and some different points that you have. And here is your lower dimension. So it's the final plot that you get in your data set. So the final dimension should, for example, if you consider point I here, the probability of these two samples being together should be the same, both in the low dimensional space and the high dimensional space. So basically what tSNE will say is that the probability of these samples being together should be the same probability also in the lower dimensional space. So this is the idea of creating an optimized way of representing your data. Andre, you have a question?
I have a question. Like, I understand the difference between the high dimension and the low dimensional space, but like in PCA, it depends where you look into space, how the image sees, like how you see the image. But in tSNE, does that also matter? If you go to the slide before, if you look at the spiral,
yes.
Like, can it be that you're looking at it? If you would be looking at it from the right side, you wouldn't see the spiral. You would just see one big cluster, as you would in a PCA. Or is that not true for the tSNE and the UN?
Exactly. So here you have the raw data is 3-dimensional. So if you reduce the space into 2 dimensions, then you have just a 2D plot, for example.
Yeah.
For the tSNE, you will still see, for example, a spiral or maybe a line that you see like all the points lined up.
Okay.
Yeah. I think that will be more clear later if I show another example.
Sorry.
Yeah. No worries. Yeah. But I'll talk about that a little bit more, I think. So the idea of tSNE and UMAP as well is to optimize what you see in the plot to what the real data looks like. That's the main idea that you should think about. So let's think this in a very didactic way. So imagine now again that you have gene A and B, and you have these 3 clusters, right? The red, the blue, and the green one here. So the first idea of tSNE is to assign those points stochastically to 1-dimensional. So here we're reducing the space from 2 dimensions to 1 dimension. So that's why we have just one line here. So first we just assign them randomly to this line. And then what we will do is try to find the distance in the original space and compare to the distance in the reduced space. So here we see that the red ones are very close together in the original space, but they are very separated in the reduced one. So that is kind of wrong, right? So we need to find, we know that there is what do we find, like a cost or error in this reduction. So this reduction does not fit with the original data. So the idea is that we then will pool those samples together. And the same thing goes for the blue one, that you'll find what is the nearest neighbors in the reduced space and the nearest neighbors in the original space. You see that they are very close together, and here they are very far apart. So we also need to force them together. And then after some iterations, we get to another position, for example, where the red ones are still far apart compared to the original data. The same thing goes with the blue ones. They are still kind of close together, but not really. The green ones are also still far apart compared to the original data. So then you can go on and update the positions of those points in the reduced space until they converge into a way that the red ones, now they are very similar to what they are in the original space. The same things for the blue ones and the same thing for the green ones here. So what tSNE optimizes is that the distance between the nearest neighbors, for example, like the red ones here, should match what you have in the original space. So you can see that the blue ones here are in the middle and the green are far apart, which also kind of represents what are the distances between them in the original space in here. But for tSNE, that doesn't matter. So you might see the two clusters swapped, and I'll talk about that in the next slide, I think. So this is the general idea, how you reduce the dimension of how many dimensions you have to a 2D representation of your data. This is exactly the same mechanism. Do you have any questions here? Because this is a very important concept that I'll mention later as well.
Does it use the information from the principal component analysis to do the iterations, or where does the information for the iteration come from?
Yeah, so in this case for tSNE, if you can run directly on the genes, on the gene A and B, or you can use principal components as well as the raw data. Okay. For this one. The iterations basically are just update the positions of the cell, compare these new positions to the raw data. Do they fit? No. So then we continue the iteration process to optimize where in the reduced space I can have a best fit to the original data.
Because here you talk about 2 genes, but it's actually like a lot of genes that you.
Yeah. You can do all of them at the same time. So here we have 2 genes, but of course you can have all the 50 principal components and reduce that to 2 dimensions only. Tui? You're muted.
Or how about the green dots to the blue dots and the red dots? Because the green dots are pretty far now. If you have 1 dimension, the green dots are pretty far from the red dots, while it's quite comparable in the original data that you can see that is, from green dots to blue and red, they are quite similar, but now it's totally different.
Yes, so tSNE only cares about the local distances. So I just want to know if the red dots are close together, if the green dots are close together, and if the blue dots are close together. And that's one of the important concepts that actually it's a very good question, because tSNE doesn't care about the relationship between the green ones and the red ones in that case, because they are not close together. So you will see that in the next slide, I will come to that as well. So this is one of the idea of the local embedding, which I only care about the closest ones. And global embedding, I only care about the closest ones and also on the furthest ones apart. So in this case for tSNE, it doesn't care about that, but on UMAP that is very important. Yeah.
Can I ask about that? When you say like it only cares about the closest ones, is that something you define? Like what is closest? Like how many are the closest, or like what distance is the closest?
Yes. So in the next slide, I'll probably answer this question now. Yes. Okay. So ask me again if I don't answer that. So there are many implementations of tSNE. The first one was called rtSNE or vSNE, also in MATLAB if you have used that for flow cytometry. And there are many hyperparameters or tuning parameters for tSNE, over 10 usually. And that can be optimized for your data set, and there is no ground truth how much you should use. One of them, for example, are perplexity, number of iterations, learning rate, and theta. And for perplexity, that actually means how many neighbors you consider. So if you have a very big data set, so ideally you will have also many neighbors to consider. In that case, you can have a higher perplexity and consider 50 neighbors. But if you have a very small data set, 50 neighbors will basically group everything together. And then you don't have this separation of the clusters. So it really is data set dependent. And those same parameters are kind of similar as well for the UMAP that I'll talk about in a minute. And I will highly recommend to play with these kind of parameters. And they will be very similar to UMAP. But check this website later in the presentation, maybe at night before bedtime, because it's really good and it's very interactive that you can choose and tune these parameters and see how is your output in the reduced dimension. And the problem is that I cannot say that, oh, this one does that, because it's kind of the interaction between those parameters that will result in an effect. So yeah, basically the perplexity is how many neighbors you are considering. Number of iterations is to let the process of optimization of the graph complete. Yeah, and I can't go into details of every single one. There are many, many more as well. And you see that a little bit on the exercises as well. So some important notes about tSNE is that unlike PCA, it's a stochastic algorithm. So every time that you run, so for example, these are 2 tSNE runs, they will look completely different unless you use like set seed in R or in Python. The cost function never reaches a minima. To say, for example, which is the best graph, there is no ground truth for that. So there is no way to find which one is the best representation for your data. So you just need to run and see if that fits. So for example, in here, everything is kind of mixed up, and here they are well separated. So as I mentioned before from Tui's question, tSNE only cares about the local embedding, so the distance between the cells that are kind of grouped together. The distance within a group are slightly meaningful, but not between groups. What I mean is that, for example, here you have this cluster in the 2 embeddings from the same data. These like pointy shape is quite relevant. It could be the differential path or some specialization process of those cells, and it's visible in both UMAPs. However, the distance between this cluster and this one that you can see, it's quite different in the 2 embeddings. That distance is meaningless. It doesn't mean that those cells are more similar to each other than these ones. So this distance doesn't mean anything. It has no meaningful information. The same thing, I can have an example like these 2 clusters. These 2 clusters are actually in here in this embedding. So they are completely random. So this is something that you should always notice for tSNE plots. And that's one of the reasons why you cannot use tSNE for trajectory analysis in general, especially if you have the clusters that are super far apart, like this one. You can, for example, use this part, but not the other ones. And one problem about tSNE, if you want to add more samples to your map, you cannot do that because you need to rerun the whole algorithm. And you see that, especially for big atlases, that is a very big issue. Lately, there is also another faster implementation of tSNE. If you compare the latest one with the traditional one, it's much faster. And I'll come to that as well later for UMAP. I don't know if this is the current implementation in Surat. It wasn't until 1 year ago. I don't know now. I need to check. But usually tSNE is slower than UMAP in general. So to keep in mind, tSNE is a non-linear method for dimensionality reduction. It was the gold standard method for single cell. And it can be run, for example, on the top principal components. But you still see them quite a lot, especially in flow cytometry. So the problems is that it does not learn a specific function for mapping new points. The cost function does not allow you to find what is the best embedding. And that same problem also happens in UMAP. And there are too many hyperparameters, and you need to find which one is best for your data set. and it does not preserve the global structure. So the distance between clusters has no meaning at all on tSNE.